First, I'll be talking about foundation species loss. This is a synthetic talk on the work that he's been doing with that over the years.
Thanks, Dave, and I'm glad to see such a great turnout here. This is really good, and the discussion goes well today. So I'm going to focus my 10 or 12 minutes on one of the pieces of LTER4, which was to look at loss of foundation species in our forests. Foundation species being particular taxa that structure ecosystems, that build ecosystems, that provide habitat and structure for a wide range of species and modulate core ecosystem processes. In LTER4, our primary focus was on the hemlock woolly adelgid system. For those of you who aren't completely up to speed on the adelgid, we got this small insect that was introduced from Japan in the 1950s to Richmond, Virginia. It has spread throughout the eastern part of the United States in the last 50 years, where it has destroyed large swaths of eastern hemlock, which is a characteristic late successional and foundation species in our forests. This is a adelgid destroyed stand in Connecticut. At the same time, people respond to the impending loss of hemlock by cutting their forests and trying to extract economic value while they still can. And so the primary interest in LTER4 with this was to begin to synthesize and bring together a combination of observational studies and experimental studies designed to understand what the consequences of the loss of hemlock would be for eastern forests, for forests around here, for forests throughout the region. We've approached this in a set of different research thrusts and directions, ranging from retrospective studies, palynological studies, historical studies, through to modeling and forecasting, and I want to highlight a few of these pieces here this morning. From the paleo record, we have a variety. Harvard Forest Force has one of the most detailed, spatially dense collections of pollen records anywhere in the world, and here I highlight examples from two areas in northwest Massachusetts and out on the Cape, showing traces of hemlock, which is the focal taxon we're interested, as well as oak, which is another key component, foundation species in many of our deciduous forests, and which is also showing interesting dynamics right now that we'll get to in a little bit. But about 5,400 years ago, both hemlock and oak declined precipitously, region-wide, not just in Massachusetts. The reason for this has been long debated in the literature, whether it was something related to climatic changes, and there's strong evidence for prolonged and excessive drought about 5,400 years ago that is coincident with these declines. There's also been some suggestion that this was caused by outbreaks of native insects, and contemporary declines of both hemlock and oak are related not only to climate changes and droughts, but also to insects. So whether drought is the driver, the follower, the precipitator of this, there's clearly a strong climate signal, and there's the potential for a biotic signal. Either way, there's very rapid and dramatic biotic responses. We've been trying to understand how rapidly these things happen on our landscape now. and how we might forecast these. And we've developed a suite of models looking at the spread of the adelgid. This is work done by Matt Fitzpatrick and a group of us here at the University of Massachusetts. This is the observed spread of the adelgid. Remember, the adelgid was introduced here in 1950, and it spread fairly rapidly, beginning about the late 1970s, first into the Northeast, and subsequently further south into the southern Appalachians. So this is the observed pattern of spread. And then we modeled it, and we said, based on climate alone, so thinking about climate signals as we might have seen in the fossil record, based on climate alone, where would we have expected to see this? And what this illustrates, what this larger map shows, is the difference between what we modeled and what we observed. And so based on climate alone, the adelgid is not very fond of cold winters, and neither are we. After this winter. And so the adelgid in a model framework would have been expected, in a strictly climatic framework, would have been expected to move south first and then north as it warms up. And so our model over-predicts the currents here early on and under-predicts the occurrence here early on, which shows us some of the limitations of strictly relying on climate models. I want to illustrate this in another way. This is a result of 350 different simulations. Each line is a simulated track of the adelgid in our model simulations, beginning with an infestation enrichment. And the large circles are the center of mass of the adelgid distribution in the eastern United States. So the model forecast would say that it should have stayed roughly in the southern part of its range until the climate really began warming, and then you start to have it being able to move northward, so that by 2008, we might have expected the sort of center of mass of the adelgid distribution to finally be getting up to around Connecticut. In fact, this track here is the observed center of mass of the adelgid distribution. It moved very rapidly north first and then subsequently expanded south. And this suggests to us, along with the actual observational data, so we have a record from Pennsylvania in 1969, and then shows up here in the 70s and here in the 80s. It highlights the importance of rare and unpredictable events in driving these sorts of patterns, as opposed to simply large-scale ensembles of climatic change. What happens when the adelgid gets to a site is it eats and kills hemlock. Dave Orwig has established a transect ranging from Long Island Sound up to the southern Vermont border, and has been following plots of hemlock now for going on 15 years, and this illustrates the percent of hemlock mortality at 3 time slices, 95, 05, and 08, at these different sites as we move from south to north. And you can see that in the south, when these transects were set up, these plots were already completely destroyed. But as we move further north, it's been moving, killing trees through time. We've set up an experiment here at Harvard Forest as well. Many of you have been out to the hemlock removal plots at Sims, where we have simulated adelgid death by girdling trees, and simulated logging activities by logging trees. And girdling trees kills trees just like the adelgid does, except there's no adelgid there, so that we're able in our experiments with our parallel controls to separate out the effects of the adelgid from simply the effects of killing the trees. When we established this plot, these experiments in 2003, and then applied the treatments in 2005, the adelgid was not yet present at the Sims tract, and very uncommon here in Peterstown. This is simply an example of a temperature trace of what happens when you do this. The deviation here is plotted relative to the control. So the log plots, shown in black here, get very warm in the summer and very cold in the winter. The girdle plots take a little bit of time to catch up as the leaves drop off. Took them about 1.5 years or so to die. But by 2.5 or so years into it, the log plots and the girdle plots have converged in terms of their temperature response and other similar responses in terms of carbon cycling and nitrogen cycling as well. The adelgid, however, has arrived at our experimental plots, and in 2009 we began, we did some large-scale surveys of adelgid infestation in our experimental plots, and these are our 2 control plots in 2009. All the hemlock trees in our control plots are shown in green. Those that are infested have the black bullseye that were sampled, and the gray ones are the ones that were sampled that were not infested. And so we're up between 1/3 and 1/2 of the trees now are infested with adelgid in these plots. And so we are now moving from a situation where we have an experiment set up where we have control plots contrasted with physical deterioration and logging. We now move into a new phase with this experiment about 7 or 8 years into the experiment, where we now can contrast the difference between the impact of the insect itself relative to physical deterioration and logging. So this experiment is playing out in ways that we had hoped it would, although we are, of course, sad to see. It was really actually quite a shock going out there last summer and seeing these trees just coated with adelgid. And we can hope that this winter sort of knocked them back a little, but we're sure that it will only be temporary. A couple of other examples of impacts and results. This is nitrogen availability in some of the plots that we've been studying, and also presages our next few talks on urban-rural comparisons. The lower sets of lines are sort of normal hemlock stands. The dotted lines are our plots out at this hemlock removal experiment. So the log plots in the green dash, and the girdle plots in the purple dash. When we apply the treatments, there's an increase in nitrogen availability as we get on our resins that reflects rapid rates of decomposition and litter fall and things like that. And the log plots, of course, go up very rapidly after they're logged, and then the nitrogen cycling recovers as these plots are now just covered with raspberry and birch. The girdle plots take a little bit longer to respond, and they showed their nitrogen spike in 2008. The hardwood plots and the control hemlock plots have been uniformly low and very similar throughout the time. These 3 solid lines here, the green, red, and blue, are 3 different. These 2, the green, I'm sorry, the red and the blue here are our hemlock destroyed stands, adelgid destroyed stands, and uninfested stands in Connecticut in this long transect. And they're running at about the same level as our plots at Sims, which is encouraging. And then these 3 very light ones here that show this dramatic peak are 3 sites at the Arnold Arboretum in Cambridge. And they show much, orders of magnitude more nitrogen going through this system in principle, because of the urban environment. These sites have now been logged and shipped because they were completely destroyed by the adelgid. So where are we going from here? We have we're looking at whether we're seeing a sort of switch from a hemlock system to a hardwood system and how abrupt this switch is. We have a conceptual model of how things work when foundation species such as hemlock are present driving the system versus when we take them away and other things drive the system. And one of the things we need to move from is a nice conceptual model to actually being able to quantify this model and forecasting where we're going. So this is an example of a forecast map that Matt Fitzpatrick has done of hemlock mortality under the A1B climate change scenario, which is sort of the mid-high scenario, and we all know that the scenarios are consistently under-predicted, and basically shows mortality by 2050 extending all the way up the Connecticut River Valley, northern New York, and into Michigan. Working with Paul Moorcroft and Marco Albani, we've looked at carbon cycling and net ecosystem productivity in these stands, showing a rapid decline as hemlock is lost, followed by a recovery as it's replaced by rapidly growing hardwoods, and again showing a similar dynamic through 2050 as the adelgid moves into the upper Midwest. Potential new directions that we can think about as we move into LTER 5. We've talked about our shift in this long-term experiment as we move to a adelgid-infested situation. The adelgid is also present on Prospect Hill, and we should be doing much more linking it with ongoing measuring and monitoring programs on Prospect Hill. Oak is disappearing on Martha's Vineyard in the Cape, and we have funding from NSF through a RAPID grant to explore that. There's an oak removal experiment at Black Rock Forest that some of us are participating in. It's very similar to that at our hemlock removal experiment, and there's lots of opportunities for future scenarios and future models.
Great. We'll time for one or two quick questions.
Aaron, there was a huge loss in the Worcester area. Any thought of linking public forest research that you're doing in Martha's Vineyard to what happens?
That'd be great. We should do that.
You had originally said that you were going to look at the mechanism by which the adelgid kills the tree, but you didn't do anything about that.
No. I can't. We actually don't know very much about still know very little about how adelgid kills the trees. Evan Cracer, who at the University of Rhode Island, and Joe Elkington at UMass, and the rest of us got this proposal to do adelgid distributional modeling. Matt Fitzpatrick was a postdoc on that, and he did these modeling things. He left for a position at the University of Maryland, and we took the last 2 years of that postdoc and have used it to hire a postdoc based at URI who's working directly and finally on the physiology question about that. So all I can say is we've had a postdoc in place now for 6 months who's actually focused on that question, but we don't have any results.
Based on time, I think we'll move on. We can have some additional questions for Aaron.